So this is one of the most popular data set to work with, iris data set. And this is the data set which will take to work now. So this is inbuilt in GRAS, but you can also get in GRAS or other databases. What we will take here is the inbuilt data set which is already present. So this data set of iris, it contains 150 records, and there are three species: iris setosa, versicolor, virginica. So this data set, it contains length and width of petals and sepals. So here petals are given, which are the petal, and sepals are also given. So you became acquainted with the data set. Now you can work with the data. So the next thing will come is manipulating data set. Now the package which we'll be using is plier, dplier. So it has a lot of functions, convenient functions which we can use. And there is one operator which is called pipeline operator, that is percent greater than sign and percent operator. This is used in this dplier for manipulating data sets, and it can do several functions like filtering, arranging data, selecting data, mutation of data, summarize data, group by, it can do grouping of the data set. So this particular package has a lot of functions, and it is very useful for data analysis. So it actually had the package called Magritter package. So both dplyr and Magritter package, both should be actually installed and called for in the library, because these works together. And then the pipe operator, it which I'll be showing you, it lets you write the functions you want to combine from left to right. So when we want to combine functions, then we can use percent greater than sign percent, which is a pipe operator. I'll refer to this as a pipe operator from later when I discuss this. So we have to install to do this, we have to install Magritter package. We have to call on the library Magritter. These are essentials for the data manipulation, which I'm going to show you. And mostly what are manipulation, why we do that? To clean the data. This just I'm showing representative what we do. We read the data, we clean the data. We have come to this point that we have read the data, and we have also cleaned the missing points or some data. We want to summarize the data, we want to plot the data, then we want to summarize. We want to do such analysis on our data, and we want to do some other manipulation. So these are essentially the steps to call on the functions to change what data from one form to another. So these are also which I've said already. I've said about arrange, filter. These are group by is there. So I'll just give a brief. What is transmutate is a function which is added computed value to the data frame and removes other columns. Only the computed value remains and other columns gets removed. Arrange is order the data in sorting columns. Filter is picking certain rows where selecting others. This we are doing using our packages. Group by is splitting the data into subtables based on column values. I'll be showing in the demo what are the things, how do we implement, what are the applications. Now the last part we'll do is tidying data with tider. So this is also a library which we'll be using to modify the data frame so that column becomes names in a factor and other columns becomes values. So we'll go through this in demonstration to find out what are the things which we'll do. And finally, I'll just recap it. What I want to present here is data frame. I wanted to show some simple arithmetic functions. I wanted to show data in the form of categorical data, find out the data shape, and tell you some useful packages which are used for data manipulation. Then missing values, how to deal with that, and what is the package for imputation in R, that is mice package, and then data tidying. And so for tidying, we are also using a package here. So I'll be showing you some useful packages which are required for manipulation. So So I'll stop sharing now and I'll stop sharing this and I'll show you the R studio. So this is our studio. I hope it is visible. Everyone can see the R studio. We come to data manipulation. Before that, I'll show you some of the data sets which are available in R. How do we see the data sets? Library data sets. When we run this, set of data sets, we'll see the data sets which are available. So these are the data sets already available in our air passengers, CO2, then from so many data sets here, UK gas, air quality data sets, which I have shown you before. These are all inbuilt data sets which are present in GRASS. Virus data sets already there. You can get from outside also, Kaggle and others, and use Excel sheet and then import it and work with it, or either directly we can take iris dataset which is present. Datasets which are present, the documentation for that. And now we'll install packages, dplyr. So install.packages dplyr run. Restart, you say yes. Along with me, you can also install these packages. After installing the package, you can either do from here or come to the package install, and from here also you can do the same thing. Install. So library is dplyr. Will also say it's busy somewhere. Wait for 2 minutes. So installing the package for dplyr, then meanwhile in help, just type in the package and what it is used for. I'm using Magritter, MAGRITTR. Just check out. So if this is getting downloaded, so if my R will become a little slow, it will get downloaded, installed and all. So meanwhile, you can also download and do certain stuff. Download the package dplyr, then library for dplyr, then install package for Magritter, and then it's installed. Okay, we'll give it time to get downloaded and installed. Library. ML Bench is machine learning, installing some packages. These packages to get installed take some time, and every time we have to install and do it. Documentation for ML Bench. A certain documentation will get already. What are these are the packages for machine learning problems. Okay. I'll show you for car and I'll work with iris data only, but it's just in order to show what are the data when we say cars, what are the applicable things. Cars. Okay, let's see. This is the data set of car. Such data set doesn't contain any missing values, so you can directly work with it. But when you have a data set containing missing them, go to the previous section where I've shown you how to deal with it. You can implement those parts. It's car, the header. Want to just see the Q plot. I think ggplot is not there, so it's not that. We need to install ggplot2, then only these plottings will happen. So in case here it is not there, we'll not be able to plot. I'll show you later. I think in data visualization, ggplot2 is being shown to you. So these um, packages and this library should be there in order to run certain commands. Otherwise it will show some error. So when you see the error, then you have to go to package, then install, then ggplot. If it is there, then you have to install the ggplot. Library ggplot. So this plotting is shown. So if you want to use certain functions such as plot, qplot and all, you should install the packages of ggplot and call the library. Otherwise it will show an error, then you will not be able to know why the error is occurring. So better be acquainted with data set or which function needs which kind of packages. So just note down and keep it aside, like what kind of packages are required for your data analysis, what you want. So this is the Q plot. This is for tail 3. From end, 3 values are being shown. This is the summary. Summary, uh, minimum, first quartile, median, mean, third quartile, max. These are shown, which I've shown you before also, that how to obtain those summary, and from summary, how can we get useful information about the data. So this is our hard data set. You can do the same thing, just experiments with different data sets when you practice. Now I'll come to iris data set, which I've shown you, and with the inbuilt iris data set, I'll be doing some of the data manipulation, which you can learn and then implement it. So working with iris data set. So data is iris. We'll run the data set. I'll show you the data also here. So the data consists of sepal length, sepal width, petal width, and species. Now first, working with the data set, we'll get the summary of the data. For sepal length, there is separate minimum, maximum, mean, median, first quartile, third quartile. There is for sepal width also these separate data sets. So we are getting information individually, separately here. Now we are seeing the names, like what are the header value separately to think the data is not run with the last data set is coming anyway. There's some error. Let's, if there is some, we'll call the library function again. Okay. So this actually function has changed in the latest mode of art, but anyway, we'll continue with this. We got a warning. So here for iris data, we wanted a output printed in tables. So we used this function. And while using this function, we got the output in a table, like sepal length, sepal width, petal width, and species. These are all printed in a table. Now we want to pick or select columns. See, there are various ways to do it. Previously, I have also shown you how to select columns, how to eliminate columns, or how to just in maybe multiple columns you eliminate, multiple columns you select. These are there are different ways. So I've shown you the ways to do that. But using these kind of packages, how to work with that and doing it more swiftly, this is another way to do it. So here, using these packages in data manipulation, you can also pick or select one column, another column, several columns. So I'll be showing you this also, taking iris data. So we are taking this operator, pipeline operator, and taking this, we are selecting petal width, only petal width in the data sets, and only 3 of it will be shown here as we run it. So this petal width only we are taking. We are picking up only single column here. This is petal width. Now, if you want to pick up several columns, several I've written, but I have only specified 2. You can give 3, 4. There are only 4 columns here. So if there are data with a lot of columns, you can just give more number of columns here. Ranges of column. Now, ranges of column here. When we see the data, I'll just go to the data. If you see the data here, you see sepal length is first column, sepal width is the second column, petal length is the third, petal width is the fourth column here. Are you able to hear me? I mean, if I'm speaking, if everyone is hearing me. Am I audible? Are you able to see my screen?
Yes, ma'am. All is well.
Okay. So this is the data set. Just go through the data set, be acquainted with it. If you have used it before, it's okay that you know what the data set contains. This is iris data set. I'll go with this data manipulation. Now we have picked one column. We have picked 2 columns. Now we want to take ranges of columns. So we can just use a colon in middle of it. So from sepal length to petal length, whatever it is present, those all columns will be selected here. So this gives a range of columns. Select one column based on column names. Now, this is more advanced level. We were doing elimination on the basis of columns. Now, just by names, we are doing some critical analysis. Now, just if we want only petal, we don't want to include sepals in our analysis, or only sepals, we don't want petals to be there. Just with the help of column names, we can segregate it. So this is the command for that. We are all using pipeline operators, dplyr, and packages so that this can be done very easily while dealing with the data. Now select column based on column names. Just run it. So from it starts, sorry, it's column based on column name. So it's petal only. So petal length and petal width. So in place of petal, if it is sepal, then sepal length and sepal width will come. Now iris data which ends with width. So length will be excluded here, only width will be given. So we'll run it. Sepal width and petal width. So just on the basis of column name, we can just segregate our data set, find our data frames and work with it. Now, it's also given that select any name that contains a T except if it is the first or last letter T. So for that, select matches within quotes, double quotes, T. So it shows all the column which has T in it, but not the first and the last letter in between. Everyone had a T. Now, we had before seen to select the columns to include. We wanted to include certain things, so we have selected. Just to exclude the column also, we can do the same thing. As I've shown before, it was not with the help of any function, but minus C when we had did for multiple column, then those columns were excluded. When it was C, then those columns were included. So minus actually excludes the thing in R. So when we say minus starts with petal, so it will exclude what we are asking R to exclude. When we are saying that exclude petals, then only sepal remains. This is what we are asking R to do, and this is what is happening. So till which one I've done till mutate, I haven't reached to mutate. What I'll do now is I'll copy and paste these codes, and then I'll give you time so that you copy and paste, because then I'm telling you options to do this. You have this command and all, so that with the help of the command you do yourself, and you see how you are able to exclude or include certain columns. So that will develop your interest in R more, because you'll be practicing it yourself now. So I'll just copy and paste. Okay, I'll stop sharing, copy and paste till now, and you perform these things, and I'll move ahead. So I've given you these things. You just have to copy and paste and do. Okay, okay. Before that, I have to give you the package and the libraries also. I'll give it now, install and do it simultaneously. Then it will be helpful to you also. Wait, I'll just give the libraries. First, run these install.packages libraries, and then go to the codes and run it. Otherwise, the codes will not run for you. And since I've given you, I'll give you 5 minutes to do it. I'll explain you why. Um, First, the install.packages are there. Install will take some time for downloading and installing the packages. Then come to the iris data, and then look the summary and select certain columns. It will not take much of the time because you have to just paste it. But installation might take some time for these packages. I'll give you some 2 minutes because I have also other data sets to perform on, but easily I think it will be done. I'll just revise some of the things which I've shown. Here, with the help of these packages, we have selected columns, one column first, then we have selected several columns, then we have taken ranges of columns, then we have selected columns based on column names. If you wanted column names based on petals, we have used start with petals, then start with sepals if you want only with sepals, ends with you can say width. If just you have all the options to do data manipulation to just extract out the data you want. Uh, Even we have contains. If it contains certain things, then also it is there. If we it matches certain alphabet, then also we can extract. We can remove it based on minus sign. For selecting, we can say start with petal. For removing it, minus starts with petal. So it removes a particular thing. So when you people say to me, like please signal that till now it is clear to you, then I'll be starting with mutate of data manipulation. Uh, Share the codes again. What is the problem? Screen is not visible. Okay, in chat, you just check in chat. It will not be, if you open the chat, all the codes are there. Just check and let me know if you are able to see the codes again. I can copy it again if you want, but it is still there. If you see and check in chat, you'll get it. Because I have pasted the code there, I have actually not shared the screen right now. So please check the chat window, you will get all the codes. First, use the library install packages, run that in R and then come to the codes and then run it. I hope it is clear. You will not be able to see anything in the screen. Okay, chats are cleared. Okay, fine. If it is cleared, I'll again copy and paste.
No, no, it is visible, ma'am, still.
Oh, it is
The type of
visible.
the script is there, yeah.
Oh, but Pallavi is telling that she's not able to see it.
What you can do is, if it is in a text file, you can share it. There is an option, file sharing. You can share it. So everyone will get that.
Okay. Text file, so it's not there. It is in R.
Oh,
Anyway, Pallavi, I'll be sharing the R. Even in the chat boxes, codes are not visible. Anyway, I'll paste it again in order to see if it's visible to you or not. Just to see if I'm pasting if it is visible. Let me know if it is visible, huh? Because then I'll be not.
madam, we have posted it in the host privately. So that's why it is not visible. Please
Okay.
to
Sorry,
attend it,
sorry.
all attendees, then it will be visible.
Okay, fine. It was in default mode. Okay. Okay, these are the uh, functions. These are the libraries and the thing. Okay, I'll give you the other things. I think half of it is done. Anyway, see, I'll be sharing the thing also and just try out some of the codes so that you do it along. what is left out and paste it. Everything is pasted. I think it's all visible now. The libraries are given, packages are given, and then the commands are given. So you can practice it along. Share my screen now. Okay. Till this part, I have shared the code, like to select a column with the name or with the first letter, last letter, in between letters, ranges of column, the basis of column names. This till now I have shared the code. I hope you have followed it till now. And please let me know if you have followed, because I'll start with mutate then. That is also one of the type of data manipulation. Mutate, what we do, we add computed values to your data frame. It adds a column to the data frame by specifying an expression for computation. Now, because I've given the data, this codes and all, I assume that you will do, because I'll explain this again when I give the code in the comment section or in the chat section. Then I will give you some time to copy and paste the code and see for yourself. And for now, you just listen to my explanation, and after this I'll share the code and then practice yourself. Just for 5 minutes, I'll talk about the data set, and again I'll give you the data set to practice for yourself. And then from scratch, you try out and see how these commands and functions are working for the data sets. So here, when we use mutate, what we are doing, we are adding a column to the data sets by specifying the expression for computation. So what we want now, we want petal width plus length. Let's see the data first. Now we want petal length plus petal width as a column, single column. For this, we want to use mutate. Everything else we have tried out. With petal, we have selected only petals, sepals with letters, or with only ends with width or ends with length. We have separated and these things and tried out several things. But now when we want to add both the columns and want a single column, for this we need functions such as mutate. So go through the data and check out what you want to do for data manipulation. And first, what we are doing, we want to add petal width plus petal length, and we want this to be named as petal width dot plus length. This is the name of the column. Now let's run it, see how it runs and what are the outputs we get. Okay, so we are getting only one column right now. The column which you are getting is addition of petal width and petal length, and the name of it is petal width plus length. So this is the name, and only single column we are getting in mutation by using mutate. Now we want to add more column. We want to use mutate function, petal width with petal length. We want to add sepal width with sepal length. So both of these we want to add. We don't want separate values, but we want to integrate petals together. We want to integrate sepals together. So for this is the command which we use, mutate, and within brackets, we are giving the name of the column which we want. We are giving the operations which we are trying to perform, and this is to add more columns. So we'll start with the mutation. So here we got the sum of sepals and the sum of petals. So these two columns we required and we got, and what functions we used was mutate function. So this is the use of mutate. So when you want to do certain things, such as adding two columns together and get you want only the output of those columns, use the mutate function. And I'll come to transmute now. Transmutate. Mutate was a function, now transmutate. It is mostly like mutate, except it combined with a select so that the result is a data frame that only contains the new columns. Okay, let's try transmutate. Run. Only it contains the same column. For others, it doesn't contain. So specifying transmutate and mutate actually is because of the function it does. So if you want only one column, use transmutate, otherwise use mutate according to your need. And then comes arrange. But before that, I'll just give you code for mutate and transmutate. And if not others, just do this thing that you take this and apply this on your data set so that at least you move ahead and practice a little bit of R. I have to stop sharing to copy it. Copy paste. Okay. This is a like you have to copy little longer codes are there, but anyway, copying this and doing this will make the functions very clear. We're getting to know, okay, this is a function for this particular application. So while I've given you the data sets, it is already inbuilt. All the functions and the codes are already there. So copy and paste and see the results yourself. It's pasted. Everyone is able to access, everyone. Just let me know if you are able to see the code so that I know you are able to see and copy and do it in your own art. Okay. Okay, thank you. Okay. Give you some time to that you run the code and see for yourself. If you're able to see the code, run it and see. Divide pair functions to select one column, several columns, ranges of columns, then also columns on the basis of names. These are the things which we have covered. Then we have come to mutate. By mutate, we are adding certain columns together. In transmutate, what we are adding, we are getting only that column. So while performing this, you will be able to see the difference, what these codes are giving as a result of it. And once you finish this, I'll come to arrange. Okay. Anyone can let me know if they are doing it, or after waiting for 2 minutes, I'll start with arrange, because there are other functions also which I have to show, and you can access it easily through the chat and yeah. I've shown arrange, then there's filter function to show, and what else? Logical expressions, group by. Okay, while you doing, I'll start with arrange. Arrange is also a function in this package. So it reorders the data frame by sorting columns according to what it is specified. You can stop doing what you're doing, because copy and pasting you can do later. You just know what arrange function does. It reorders the data frame by sorting columns in order it's specified. If we are arranging in place of sepal length, let's run it and see what is the function of this. Okay, increasing order 4.3, 4.4. The basis of say septal length, others are not taken into consideration. So this is basis of sepal length, this is increasing order. Uh, but if you want in decreasing order, we have to give the function arrange under bracket decreasing DESC. So when we give that function DESC, then what we get is a decreasing order of sepal length. So let's run and see. If it's not taken, just save the whole thing and then run. Okay, what will it happen anyway? There's some problem. I'll just stop sharing and I'll start my R again so that it runs properly. While you people copy the code and paste and try running the codes, I'll stop sharing and run R again.
Madam, you can unmute.
Okay, I can't share. Give that permission.
Network issue there?
Okay, thank you.
Is there any network issue, ma'am?
No, suddenly it got disconnected.
Okay, okay.
No, network is okay, but how it got disconnected?
Fine.
At time, I think what codes I have shared was being practiced, so I'll move ahead. I think because I've restarted R, I have to install the packages again. Those who did not follow the installations can check out install package UI player currently installed again. Install packages. Arrange. So in case these functions are not running, you can try reinstalling the packages, or in case again R has been shut down, then again you have to install these packages and again call the library function. So be ready for that in such cases of shutdown of R. Just check if it is running. Remove certain things. Let's say date. Okay, it's become slow, but it's working. So I'll just tell you each of the function, meaning why we are doing such things. Arrange. When we do arrange, that's arranging in increasing order. When we use DSC, it is arranging in decreasing order. When we are doing filtering, here we are filtering the sepal length which is greater than 5. So that filtering is done for sepal length which is greater than 5. It is being filtered. So all these function has certain filtering effect, like when we arrange, we are doing arrange for filtering. We can do it greater if sepal length we want greater than 5 or less than 5. We can just specify it and get the output. So similarly, petal length less than 4 also we can change in the expression here to get a separate output. In logical expressions, what we are getting, if like here in filtering, we are taking only the sepal length which is greater than 5. We want the sepal length greater than 5, but we want the species to be virginica. We don't want from any other species. In iris data set, there are different species which are present, but virginica. From only this species, we want the sepal length which is greater than 5. For no any other species, we do not want such data sets. So for that, what we do is we put logical expressions, and in logical expressions we give both filter plus equal to, that is logical function species equal to virginica. Only if it the conditions satisfy both, then only we select this species. So with the help of logical function, we are getting the species virginica 6.3, 5.8, 7.1. This is the species we wanted, and we wanted the sepal length also be greater than 5. So this logical expression has given us the required results. Uh, in case you run and there is some problem, you can restart R, as I was facing some problems while starting. And if there are some errors, you go ahead and again install the library, then I think the functions will properly work and you will get the results. In case you are stuck somewhere, then this is what you can do. Now group by, okay. This is also one of the data manipulation process. Group by is splitting the data into subtables based on column values or into some other type. Now we want to split the data according to species. Okay. Now when we want species name, then how we do it? It's group by species. Now the species are what? Setosa species and other species we have to check. Only 3 are given. So these when we group by do species, then only the species are only taken as a table and summarized. We'll see the summary in mean dot petal and mean dot sepal length. If we want only the mean, then we'll do this summarize. Let's see. So we get the petal length and the sepal length mean. So I'll just give the data sets to you so that in case there is some problem while running the data set, you can look into it. I don't think there'll be any problem. Everything is already, the code is run, but in case there is some problem, you just retry it. And I think all the codes will get run, and call the library again in case it's not getting run. So till group by, I'll give you the code and you practice till group by, and I'll start with this splitting the data into different groups and compute the summaries for each group. So already we have done for one, and for others we'll do the summaries. Then summarize observations. So let me know, did you get the point why we are doing this? We have some functions already where we don't need packages. We can select the columns, we can deselect the columns. But here the advantages are that if that only by petal name or just with the help of a letter also we can filter out. Just we want only sepal length greater than 5. You want some values which is greater than 5 or 10. You can easily filter it out. You want certain species only, you can easily filter it out. And you can easily add two columns together and get only the resultant column. So these are very easy and quick ways to get the data which you want to do analysis on. So these kind of packages which dplyr and all these are so useful because of this utility which it is providing. And that is why, because of the utility, we are using these packages while doing the analysis, while showing various applications of these packages and the library. So I think group by is still what I have explained you and I've run. I'll just give this data again. I think I'll stop sharing, otherwise I'll not be able to copy paste. So I've copied and pasted. What I've copied and pasted are filtered functions. How do we filter a certain kind of data if we want a column to be less than certain things, maybe the range in column to be less than 10 or less than 5, we can use the filter function. If we want only certain species data and a sepal length greater than 5, then we can use logical expressions and also filter functions in that. If we want to group on the basis of sepal or petal or any other characteristic, use group by function. And different functions has different utilities, different ways which it can be performed. So just copy and paste and see what are the results you are getting. I'll share my R again since I could not copy while I was in share mode. So I've given you the course. So please practice. I'll go ahead now. Till group pi, I have already shared the code, and it was like summarizing mean petal length and summarizing mean sepal length and getting the result. We can use groups in different ways to split the data. How we want to split the data, we can just give summarize and we can get different results. So there's several ways to use grouping, depends on the use you want to do. And here I'm using iris data set, hugely popular data set to do data analysis. Please take another data set also to do some analysis and see what kind of results you get. You already have the code, so you have to change the data sets and the column names and see like how the characteristic changes, what kind of information you get using these commands. I think you've all got. I just copy it and see. Now I want to go ahead because I'll be showing you the data into, yes, splitting the data into different groups and compute the summaries. Okay, let's change the group, split it, and then compute the summary. This is the command for it. Now we have three species, Setosa, versicolor, virginica. Now we have got the mean petal length using group by. We are grouping it into species. Three species it's getting grouped into, splitted into, and we want the mean. So easily we can get the mean of petal length in these three species. Now this iris observation. So number of observation is 150. We can also see that in data set. We have already opened the data set of iris. 150 is the number of observation on only 150 data sets we are doing our analysis on. And in case of categorical data, species name is a categorical data only. So the analysis which I have shown you before, you can also take iris data set and you can just do some histograms and other relevant things to know more about the data here. When combined with group, let's take a look at it. Okay, it's the number of species in each group. Setosa, versicolor, virginica. So this is all group by functions. By group by function, what all information you can get. Some of the utilities. There are more, I'm sure there are more utilities. I'm showing you a few of it. Summarize statistics. This is also to summarize the basis of mean petal length. Okay, so in 50 species of Satoka, the mean petal length is 1.46. In versicolor, mean petal length is 4.26, and virginica, mean petal length is 5.55. So, just by summarizing it, we are getting a lot of information. We are seeing that in virginica species, the petal length is the largest, while in Setosa species, the petal length is the smallest. So, these are the functions and utilities you can help you in data analysis. And next is iris data set with data points in different shapes according to species variable. Let's do some graphs also, some data visualization. We have such a data, we have grouped it, we have filtered it, we have done various things with it. Now we'll make some graphs and see the data sets. Let's see how the data set looks in a graph. We'll visualize the data set a bit. So in ggplot, if we haven't installed, we have to install again. It's installed. So here we are giving different shapes to this species. Setosa, one, the shape is different. Versicolor, the shape is different. Virginica, the shape is different. Petal width is varying. Petal length is also different in these species. So this is the visualization. Qplot call to create a scatter plot with colors determined. Okay, if you are doing the Qplot, make sure you have ggplot2 package installed and call in the library for ggplot2. Otherwise, it can show some error. So while you figure out the error, do this together. Again, okay, now the same error I'm getting, ggplot not there because the jar got closed. So again, I have to install ggplot2 and call in the library. Packages installed. Install. And only I'll in the diagram. Let's make sure you have the installed packages and libraries, otherwise these visualizations won't be possible. So here we are visualizing the petal width of Setosa versicolor virginica. And Setosa, it has the minimum petal width and maximum is virginica. We have also seen this with the help of group by. When we see the mean also 50 species are there in the data set, and in mean also it shows that Virginica has the greatest, highest width and length. I think it shows in the mean also, and also in the graph it verifies the same. We are just making the red color, but it doesn't make us easy to recognize. So giving different colors are only much better visualization technique than give a single color which are not recognizable. Facets. Okay, these are plots which we are get together. Facets are subplots showing subsets of a data. Now we use facets to see the different subset of a data in a single data plot. So let's try this. This is a very big script, but anyway you'll be given so you can copy paste to see. This is a big script to see facets where subplots are seen in a data. So this is the data subplots available during facets. I'll be giving you the code so you don't have to write so much. Color. Let's try changing some color. Do not gather function. It is which function? Okay, we need to again see which is the gather function I have to change. So these are for data visualizations. Basically, what I'll do is I'll give you the script so that you can practice and do, and I'll go for data uh, typing so that this is done. So from where I have to give the codes, I'll just give the code to you facets. Okay, I'll just give you these codes while you practice for some time. I'll be explaining you data typing later on. After you are completed with this, I think visualization is not that much required. You can later see, but I'll give you till group 5 summarize so that at least you see the data manipulation, which kind of manipulation can be done. And with the help of ggplot and other functions, you can easily see or visualize the data sets. You'll be able to see summarize the observation group by species, summarize the total number of species or number of species which are grouped, and then you can find the mean of each groups, and also just the last one is the shape map of different species, so that with the help of shape map you can come to know like which species has more sepal length or more petal length, and this visualization can give you a lot of information. Just by seeing you'll get a lot of data information by simply doing these visualizations. So when I've pasted, I'll give you some time, maybe 2 to 3 minutes while you paste and see the data sets. And be sure to install so much of packages, some of the packages which are given. If these packages are not there, then this commands won't run. So packages are vital for running of these commands. So first install, call these libraries, and then go for these codes. Let me know if you are able to access the codes, you are copying it and doing it. Whichever packages are given, you try to install, otherwise some of the functions won't run. Since I had to shut down our twice or so, some of the packages are not installed, so maybe some functions were not running, but if you do it, all the functions will run properly. May I get some response? You're able to access to the codes, you're practicing it, because after this I'll go to the last phase of data manipulation. Okay. Okay, there are some people who are able to access the code. I hope you are doing that in R also, practicing that in R. I'll give you 2 minutes while you people practice at least 2 or 3 codes you run. There is no need to run everything right now because I'll proceed further to the next step. So 2 or 3 you just pick out and you run it and see how it goes. Last part which I'll be dealing it with is tidying of the data. Data tidying is a standard way of mapping the meaning of a data set through its structure. A data set can be messy, so tidying depends on how rows, columns, and tables are matched up with observations, variables, and types. So we are using a library called tidyr, TIDYR library, and it has a function gather. So when you use the function gather, make sure you have this library. So in my last code, it wasn't there. So I had to install that and then actually run the function. So make sure that you know which function goes with which library so that those library and those packages are already installed so that you don't get any error while running those functions. So it has a function gather in tidier that modifies the data frame so columns becomes names in a factor and other column becomes values. So we'll go through this package and see how this works. So this is the last step of data manipulation I'll show. There are a lot of codes which are there in data manipulation here, which I'll be sharing. Already I've shared and already shared. So if you are using, you'll be getting a lot of graphs, visualizations, packages, and a lot of good visualization graphs from R, and this will be good for your data processing and analysis. So try out these graphs and packages in order to have more attractive graphs and visualization. So the last one I'll come as data tidier. So it includes packages, which is one package is tidyverse, the other is tidyverse, okay, and other is library. Tidier and other is tidyverse. So you need to install these packages in order to run these commands. So here also the data set which I'll be using is iris data set only. So assuming that you have got the codes and you have at least run 2 or 3 commands, no need to run everything right now, you can do it later also. So just some practice along with me. And I'll come to the last section is we'll take the data iris data set. So this is the iris data. We'll run that. Already it is open here. We know about that iris data set. What is the column names? What are the characteristics? We are acquainted with the data. Now tidying data with tidier. So we are not using this gather right now, so we can use this function. So petal length we have got. Then we are getting some blocks. Okay, again ggplot we have to get. So ggplot. Okay, I'll come to a direct tidyverse package so that you install those packages and library, and we can start with the gather function and see what happens if we use such functions in the data sets. Install. So it is successfully unpacked, downloaded, library tidyverse. Then tidy. So we have installed the package tidyverse. We have called the library tidier and tidyverse. Now we'll go to the gather function and see what it does. Measurement. So what it does is vary some species column against petal length column. So it is giving the petal length measurement 5.1, 5.9 in a increasing order. Here Q plot. Here the species are taken in a box plot. We are able to see it in a box plot manner. Attribute measurement sepal length, comma sepal width. Just taking sepal length and sepal width will plot this. 5.1, 4.9, 4.7 for these species. Then facets box plot. So box plot which is obtained is of different species, the different 3 species, Setosa, versicolor, virginica, and the box plot is based on sepal length and sepal width. If we change the attribute to petal, it will come as petal length and petal width. So these are various forms of data tidying, and these are the various outputs we can get. So there are various ways of data manipulation. I've shown you a few, and these are the things which you can do by installing packages. Without packages also there are manipulations which you can do, and you can extract columns and all. But for convenience, try doing some package installation, and there are the easy way to do it. Easy way because easily you can extract those data which you want, and these codes are also available. So I think you can go ahead with these installation, and you can go ahead with running those codes. So data tidy. I'm giving you the codes right now. I'll stop sharing and give you the codes, and I'll wait till at least this part you finish it off and tell me that some part you've completed. And what I want to tell also that in help, where I'll just show you again. Like there is some package which you don't know much about, like tidyverse package. Just type tidyverse and read about it. You don't have to do anything else. There are useful links also. Want to read more about the package? It's already there. And some other packages also which I've used here, which that time. This is for data manipulation, filter, remove, mutate. So many applications you can do here. Let's see that. It offers a set of operators. We have already used the operators and calls the function, and it has a pipeline of operator pipe, which I've already shown you. This is the sign of the operator through which we could do a lot of data manipulation, and how handy it was for us to write codes along with this operator, and great, I mean, some good results could come, and it's a good way of doing some data analysis. So use these packages, and if there are simpler data analysis to do, then which I've showed you in the first session will only come handy if simple data sets are there. If missing data sets are there, of course, then you know that second part which I've shown how to deal with missing data sets will come handy in data manipulation. And if you have data sets where you want to extract various kinds of data based on their first name, last letter, or something, then these data sets are of course mutating or combining the columns together, and these kind of things, then these packages will come very handy. And try using these packages, and whichever help you require in the documentation, you can read about these packages. There are various helps in R, but in documentation also you can get a lot of help. And as you have seen also, a lot of links are also given, useful links are given. Just you can read those links also. These packages, what are the utility, what are the use. So I think I'll stop sharing right now, and can you tell me if you got this data transfer chat. I have pasted it. Let me know if you are able to see. This is tidying data with tidying data. Tidying data. Let me know if you are able to access, because then I'll know the codes are available to you and you are doing this part. And available, at least try 2 or 3 codes, because this codes will remain, so no need to try all at the same time. Try 2 or 3 codes and see the results. And whenever you come across any data set which has missing values, or you want to just take out one column, work on it, use these functions, use these applications to or data analysis based on this. And other kind of advanced analysis is also already taught to you, and you are going to learn tomorrow also. So at least data cleaning part is done. Okay, good then. You can copy paste in R and just see some of the results. I'll just share my slides and I'll share tidying data. I just finished. So I'll just recap what I have covered. Data frame, I have covered. I've shown how to do it manually, also typing in R, and also through Excel sheet, CSV, read dot tables in Notepad. Also, you can do. You can do simple arithmetic operations in R. You can do categorical data and data shape analysis also. Categorical data which are present in R, how it should be dealt with, how you can work with that. Even in case of missing values, these are also numeric and categorical. In case of categorical data, in missing values, you need to deal with that separately. You cannot take it as the same as other numerical data. Also, take that into consideration and use the R packages which are there for your convenience. Some of the data sets with simpler analysis may not be required, but in some of the data sets, you can use these packages. And imputation is R is because it'll for dealing with the missing values. So use these imputations because if you omit omit certain rows or omit certain columns and make the data shorter, then it will not help you in the analysis. And this kind of packages are meant to help you in making your analysis much better. Data tidying and data manipulation was what I covered at last of the session. And use those packages which are being referred, or if not required, use the simpler versions for simpler data sets. So thank you.
Thank you very much, ma'am. I request attendees, if any questions or any queries, you can ask now or you can raise your virtual hand so that we can unmute you. Madam, can you please keep your last slide?
Last slide.
Yeah, contact details so that they can take a note of that for any queries or any doubts. Contact details, yes, this one.
Okay, this is for triple ITP. I'll show you the first slide, my mail ID. Anyway,
Okay,
this is
that will be helpful. You can share in the attendees chat box, ma'am, to attendees.
I'll show you. It's my mail ID is already there in the first slide. I'll show you there. Oh, it's written.
Yeah.
My mail ID. I hope everyone is able to see the screen and this.
Ms. Akshata, if you have any feedback, you can give. Akshata G. Bhatt, Deepti V, Meenakshi.
No, 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 no. ಎಲ್ಲ ರಾಂಗ್ ಹೋಗಿದ್ದೀರಾ ಇದೇ ಮನೆ ಅದೇ ಮನೆ ಸರಿ
Hello.
ಅಡ್ರೆಸ್ ತೋರಿಸ್ಬೇಕಂತಂದ್ರೆ
There are no questions, ma'am, from the participants as of now.
Okay.
We'll get back to you.
Okay. There's a mail ID on. Please feel free if any queries are there. Thank you all.
Thank you. Thank you on behalf of KST and on behalf of all the participants and on my own behalf. Thank you very much, ma'am, for today's data management using our software. I also thank Professor Uttam Kumar sir, who is a key person bridging this workshop between IIITB and KSTA. Thank you very much, sir. And also thank you all the participants. Tomorrow we will be sharing you the link, different link. Now I will be sharing the joining link in chat box. You can copy it and save it in a notepad, and tomorrow you can use the same thing. Okay. Thank you. Thank you, Anand. Thank you, ma'am, once again. Bye. Thank you, sir. Uttam, sir, thank you very much.